Hello everyone and welcome back to Heal Hub Edu. I'm Dr. Amer and I'm delighted to have you back for part 2 of our Neuroembryology series. If you haven't watched part 1, I highly recommend doing that first because today's lecture builds directly on the concepts we covered earlier. In part 1, we learned the foundation of nervous system development. We discussed gastrulation, notochord formation, neural plate, neural groove, neural tube formation, neural crest cells, primary and secondary brain vesicles. Let me remind you of one important concept. Everything in the nervous system begins with the neural tube, I repeat. The neural tube is the embryological foundation of the entire central nervous system, including the brain and spinal cord. Now, in part 2, we'll build on that foundation. Today, we'll explore development of the spinal cord and the embryological development of the forebrain and hypothalamus, development of the midbrain and hindbrain formation of the cerebellum, development of the ventricular system, flexures of the developing brain, cranial nerve development, meninges, cerebrospinal fluid pathways, important congenital anomalies, and high-yield clinical correlations. By the end of this lecture, you'll have a complete understanding of neuroembryology from the earliest stages of development to the formation of the mature nervous system. Now let's bring everything together and understand how the spinal cord develops from the neural tube. By now, we know that the neural tube is the embryological precursor of the central nervous system. But how does this simple tube become the highly organized spinal cord? Let's find out. Initially, the wall of the neural tube is composed of rapidly dividing neuroepithelial cells. These cells multiply continuously and eventually organize into 3 distinct layers. Remember this important point. The wall of the neural tube differentiates into 3 layers. Let me repeat that. The wall of the neural tube differentiates into the ventricular layer, the mantle layer, and the marginal layer. Let's understand the function of each layer. The ventricular layer is the innermost layer. It contains stem-like neuroepithelial cells that divide to produce neurons and supporting glial cells. It also gives rise to the ependymal cells, which line the central canal of the spinal cord. Moving outward, we find the mantle layer. This layer contains the developing neuron cell bodies. As development progresses, the mantle layer forms the gray matter of the spinal cord. Finally, the outermost marginal layer contains growing nerve fibers, or axons. This layer eventually develops into the white matter. Here's an easy way to remember it. Mantle means gray matter. Marginal means white matter. Now let's discuss one of the most important embryological events. A longitudinal groove called the sulcus limitans divides the neural tube into 2 functional regions, the ventral basal plate and the dorsal alar plate. The basal plate develops into motor neurons, which send signals from the central nervous system to muscles. The alar plate develops into sensory neurons, which receive information from the body and carry it to the central nervous system. This is a classic examination question. Basal plate equals motor. Alar plate equals sensory. I repeat, basal plate forms motor neurons, while the alar plate forms sensory neurons. But how does the embryo know which side becomes motor and which side becomes sensory? This is controlled by signaling molecules. The notochord and the floor plate release sonic hedgehog, or SHH. SHH stimulates the development of the basal plate, leading to the formation of motor neurons. On the other hand, the roof plate and the surface ectoderm release bone morphogenetic proteins, or BMPs. BMP signaling induces the development of the alar plate, resulting in sensory neuron formation. Now, if we look at the mature spinal cord, everything makes perfect sense. The gray matter, derived from the mantle layer, forms the butterfly-shaped center. The white matter, derived from the marginal layer, surrounds it. The dorsal horn receives sensory information. The ventral horn contains motor neurons. And the central canal is the remnant of the original cavity of the neural tube. Before we move to the next slide, let's review the 5 key concepts. The neural tube forms 3 layers. The ventricular layer produces neurons and glial cells. The mantle layer becomes gray matter. The marginal layer becomes white matter. Basal plate is motor and alar plate is sensory. If you understand these principles, you'll understand not only spinal cord embryology, but also the organization of the adult spinal cord. Now let's understand how the embryonic neural tube transforms into the spinal cord. At first glance, the neural tube appears to be just a simple hollow tube. But during development, it undergoes remarkable changes that eventually produce the highly organized spinal cord we are familiar with. Remember this important point. The spinal cord develops from the caudal portion of the neural tube. Let me repeat that. The caudal part of the neural tube develops into the spinal cord, while the cranial part develops into the brain. Now let's follow this transformation step by step. Initially, the wall of the neural tube is made up of rapidly dividing neuroepithelial cells. These cells continue to multiply and gradually organize into three distinct layers. The innermost layer is the ventricular layer, also known as the ependymal layer. This layer is the birthplace of almost all neurons and supporting glial cells. It also surrounds the central cavity, which later becomes the central canal of the spinal cord. Next comes the mantle layer. This layer is rich in developing neuron cell bodies and eventually forms the gray matter of the spinal cord. The outermost layer is the marginal layer. As nerve fibers grow outward, they accumulate in this layer, which later develops into the white matter. Here's a simple way to remember it. Mantle equals gray matter. Marginal equals white matter. Now let's discuss one of the most important events in spinal cord development. A shallow groove called the sulcus limitans divides the neural tube into two functional regions. The ventral basal plate gives rise to motor neurons, which carry signals from the central nervous system to muscles. The dorsal alar plate gives rise to sensory neurons, which receive information from the body and send it to the central nervous system. This is a favorite exam question. Basal plate is motor. Alar plate is sensory. I repeat. Basal equals motor. Alar equals sensory. But what determines whether a region becomes motor or sensory? The answer lies in embryonic signaling molecules. The notochord and the floor plate release sonic hedgehog, or SHH. SHH stimulates the formation of the basal plate, leading to the development of motor neurons. In contrast, the roof plate and the surface ectoderm release bone morphogenetic proteins, or BMPs. BMPs stimulate the formation of the alar plate, leading to the development of sensory neurons. Finally, if we look at the mature spinal cord, everything falls into place. The gray matter occupies the center in its characteristic butterfly shape. The white matter surrounds it. The dorsal horn is sensory, the ventral horn is motor, and the central canal is the remnant of the original cavity of the neural tube. Before we move on, let's quickly revise the 5 key points. The caudal neural tube forms the spinal cord. The ventricular layer produces neurons and glial cells. The mantle layer becomes gray matter. The marginal layer becomes white matter. Basal plate is motor, alar plate is sensory. If you remember these 5 concepts, you'll understand the embryological organization of the spinal cord, rather than simply memorizing it. Now let's move to the next topic and see how the forebrain continues to develop into the complex structures responsible for higher brain functions. Now that we've understood the formation of the primary brain vesicles, let's move to week 5, where one of the most important developmental events takes place. The forebrain, also known as the prosencephalon, begins to divide. Remember this important point. The prosencephalon divides into 2 secondary brain vesicles. Let me repeat that. The forebrain, or prosencephalon, divides into the telencephalon and the diencephalon. This is one of the most frequently tested concepts in embryology. Let's understand what each of these structures forms. First, the telencephalon. The telencephalon enlarges rapidly and eventually forms the right and left cerebral hemispheres. These hemispheres contain the cerebral cortex, the region responsible for higher mental functions such as thinking, learning, memory, language, intelligence, and voluntary movement. The telencephalon also forms the basal ganglia, which help regulate movement, and it contains the lateral ventricles. So whenever you think of the telencephalon, think of the largest part of the adult brain, the cerebral hemispheres. Now let's look at the diencephalon. The diencephalon forms the central part of the forebrain and gives rise to several extremely important structures. These include the thalamus, which acts as the major relay station for sensory information, the hypothalamus, which maintains homeostasis by regulating body temperature, hunger, thirst, sleep, autonomic functions, and endocrine activity, the epithalamus, which contains the pineal gland, the subthalamus, the retina and optic nerve, which are embryological extensions of the brain, and the third ventricle, which lies within the diencephalon. Here's an easy memory trick. Think of the diencephalon as the control center of the brain. If a structure controls sensation, hormones, body temperature, or vision, it is very likely derived from the diencephalon. Remember this important fact. The retina and optic nerve develop from the diencephalon, not from the skin or peripheral nerves. I repeat, the retina and optic nerve are direct embryological derivatives of the diencephalon. This explains why the retina is actually considered part of the central nervous system. Now let's summarize. The prosencephalon divides into telencephalon, which forms the cerebral hemispheres, cerebral cortex, basal ganglia, and lateral ventricles. Diencephalon, which forms the thalamus, hypothalamus, epithalamus, retina, optic nerve, and third ventricle. Now that we've learned how the forebrain divides into the telencephalon and diencephalon, let's focus on one of the most important structures derived from the diencephalon, the hypothalamus. Although the hypothalamus is a relatively small part of the brain, it is absolutely essential for life. It regulates body temperature, hunger, thirst, sleep, blood pressure, hormone secretion, and many automatic functions that keep us alive. But where exactly does the hypothalamus come from during embryonic development? Let's understand this step by step. We already know the developmental pathway. The ectoderm forms the neural tube. The neural tube develops into the prosencephalon or forebrain. The prosencephalon then divides into the diencephalon and the telencephalon. Finally, the hypothalamus develops from the diencephalon. Remember this sequence carefully. Ectoderm to neural tube to prosencephalon to diencephalon to hypothalamus. Let me repeat that once more. Ectoderm to neural tube to prosencephalon to diencephalon to hypothalamus. Now, let's answer another very common examination question. Exactly where does the hypothalamus develop within the diencephalon? To understand this, imagine the third ventricle as a room. The upper wall of this room develops into the thalamus. The lower or ventral wall develops into the hypothalamus. So remember this simple relationship. Upper wall equals thalamus. Lower wall equals hypothalamus. Here's an easy memory trick. PRO to DI to HYPO. Think of it as a journey. PRO stands for prosencephalon. DI stands for diencephalon. HYPO stands for hypothalamus. If you remember PRO to DI to HYPO, you'll always remember the embryological origin of the hypothalamus. Let's summarize the key points. The hypothalamus is derived from the diencephalon. The diencephalon develops from the prosencephalon. The hypothalamus develops from the ventral wall of the third ventricle. The thalamus develops from the dorsal wall of the third ventricle. These are high-yield facts that are frequently tested in medical examinations. In the next lecture, we'll continue exploring the development of the forebrain and see how these embryological structures mature into the complex anatomy of the adult brain.